Okay, so welcome back everyone. I think we can get started. So in these early lectures of the course, we've been starting at the level of the individual cells that make up our nervous system and looking at what these are actually doing in our brain. We saw last week that neurons generate a kind of electrical signal called the action potential that travels down the length of the axon, and when it reaches the end of the axon, it triggers the release of a chemical signal which across the synapse that the neuron shares with another neuron. And so it's that chemical signal and the synapse that we're going to focus on in today's lecture. And so this process of chemical communication across the synapse is really fundamental to how neurons interact with each other. In this lecture, we'll see that once we know something about how the synapse operates, then we can begin to see how neurons are doing more interesting things, like integrating different signals together and effectively processing information. And so ultimately, this is what forms the basis of our cognition, so our ability to perceive the world around us, to form memories, to make decisions, and so on. And so as we go through this course, we'll see some specific examples of the role of the synapse in different cognitive processes. Another reason why the synapse is interesting is that it underlies the plasticity of the brain, or it's a key contributor to the plasticity of our brain. And so by that I mean the ability of our brain to adapt and alter the way that it works with experience throughout our life. And so in a later lecture on memory, we'll see that there are particular structural changes that can occur at synapses in our brain that contribute to our ability to learn, to form memories, and to develop different patterns of behavior. The synapse is also interesting because it mediates the effect of many drugs on our mind, so including psychiatric medication, but also recreational and dietary drugs like alcohol and caffeine. And so this image is from a study that was investigating the effects of alcohol in rats. And so on the left here is a very drunk rat, and on the right is a slightly less drunk rat. And these two rats were given the same amount of alcohol to drink, but the one on the right was also given a kind of experimental drug that blocks the effect of alcohol interacting at the synapse in the brain. And as a result, alcohol had less of an effect on the rat's behavior. And so many other drugs are also interacting with the chemical communication that is occurring between neurons at the synapse. And then finally, another reason why the synapse is interesting is that it can mediate the effect of genes on our behavior as well. And so the genes that we inherit from our parents, a number of those genes influence the function of proteins that operate at the synapse and allow neurons to interact with each other. And this is likely to be a key mechanism through which our genes can influence the development of our brain and our behavior. So as an example, one area that this has been studied is in the context of autism. And so autism is a heritable condition, so meaning that if you have a family history of autism, it's more likely that you will be diagnosed with autism yourself. There isn't any one autism gene where if you inherit that gene, you will be diagnosed with autism, but rather there seem to be hundreds or thousands of different genes that a person can inherit and which each slightly increase the chances that they will be diagnosed with autism. And a number of those genes linked to autism are known to affect processes that are happening at the synapse when neurons communicate with each other. So the study that's illustrated here is just an example. It focused on a particular symptom of autism, which is sensory overreactivity. So this is one of the diagnostic symptoms of the condition. The way that it's experienced can differ quite a bit between different people with autism, but for many people with autism, it takes the form of tactile hypersensitivity. So finding everyday experiences of touch, like being hugged by another person, feeling the fabric of your clothing against your skin as being aversive or even painful. And so here they focused on a couple of genes that had been linked to autism in humans and are known to play a role in the function of a particular neurotransmitter in the brain called GABA, and which we'll learn about later in the lecture. And so GABA is a chemical in our brain that has a role in inhibiting or regulating the activity of our neurons. And in this study, which was done in mice, an animal model, they disrupted the function of these particular genes in sensory neurons in the mice's body that are involved in detecting touch on the body. And they found that these mice developed hypersensitivity to touch and then also developed changes in their social behaviours and also anxiety-like behaviours. And so it's an example of how particular genes can impact on the function of neurons in our body or in our brain, and in some cases this might cause changes in experience, like in the experience of touch, and perhaps if that's happening early in life, it might even impact on the development of more complex social behaviours. Okay, so in this lecture I'm going to describe chemical events that happen at the synapse that allow neurons to communicate with each other, how neurons are integrating together different signals that they receive from other neurons, and then I'll also talk about some of the different types of neurotransmitters that are found in our brain. So by the end of the lecture, you should be able to describe the chemical events that allow neurons to communicate across the synapse, explain the different signals that a neuron can receive and how these are integrated, and then also contrast different types of neurotransmitters and how they're tending to exert their effects on other neurons. Okay, so we first met the synapse in week 1. So I described how it's this specialized kind of junction point between neurons where the end of the axon of one neuron comes very close to the dendrite, or sometimes the cell body of another neuron. I also mentioned that communication between these two neurons across the synapse is largely one way. So meaning that we can define for any synapse in our brain, we can define a presynaptic neuron, which is the one that is sending the signal via its axon across the synapse, and a postsynaptic neuron, which is the one that is receiving that signal. So this is a really cool image of a real synapse, so pictured through an electron microscope. And so here we can actually see the axon terminal, so the very end of the axon of the presynaptic neuron at the top. We have a part of the dendrite of the postsynaptic neuron at the bottom. Then we have this tiny gap in between, which is called the synaptic cleft. You'll also notice these little bubbles that are present in the axon terminal, and we'll talk about these more in a moment, but they're called vesicles, and they're basically little packets of neurotransmitter that are waiting there ready to be released into the synaptic cleft. Many neurons also have dendritic spines, which are these little protrusions that come out from the dendrite of the neuron. So this is a dendrite that we're zooming in on here. And it's often at these dendritic spines that synapses are forming. And so you can see here that this one neuron has hundreds of these dendritic spines, and so it's capable of forming many synapses with other neurons. Okay, so the key learning points here. Either neurons communicate with each other using synapses, where chemical signals are sent from one cell to another, and we refer to these as being the presynaptic and the postsynaptic cell, respectively. Okay, so this process of communication between neurons across the synapse involves a series of steps similar to the action potential. And I'm going to go through these steps using this diagram here. So just to orient you to begin with, what we're zooming in on here is the synapse, so the point of connection between the end of the axon of one neuron and another neuron. So at the top here we have the axon terminal of the presynaptic neuron. At the bottom we have a part of the cell body of the postsynaptic neuron, but that could also be the dendrite instead. And then in between we have the synaptic cleft. Here we also have the synaptic vesicles highlighted. So again, these little bubbles, sort of like packets of neurotransmitter. They have an outer skin, which is made up of a membrane, which is similar to the membrane of the cell. And inside they have a number of neurotransmitter molecules. And they're found in the axon terminal. And so neurotransmitters I'll talk about more later on, but basically they're chemicals that can be released by neurons and have an effect on the function of other neurons. Okay, so step one in the process of communication across the synapse is the arrival of the action potential in the axon terminal. So we saw last week how the action potential is initially generated in a neuron at one end of its axon near to the cell body. And then it can propagate along the length of the axon until it reaches the axon terminal. When the action potential reaches the axon terminal, it can cause the opening of voltage-gated ion channels that are in the axon terminal that allow calcium ions to enter the axon terminal. So that's depicted at the bottom here. So last week we talked about sodium and potassium as these ions that are crucial to the production and propagation of the axon terminal. There are also calcium ions that are present in the extracellular fluid around the neuron and particularly concentrated on the outside of the neuron rather than inside. And the arrival of the action potential at the axon terminal can trigger the opening of channels that then allow those calcium ions to enter the axon terminal. These calcium ions have a specific role at the synapse, which is that they're able to interact with the vesicles that are found within the axon terminal and cause them to fuse with the membrane of the cell. So that's depicted at the bottom here, where we have a synaptic vesicle fusing with the membrane of the cell. And as it does that, it releases the neurotransmitter molecules that are contained within the vesicle into the synaptic cleft. So the key point here is that when an action potential reaches the axon terminal of the presynaptic cell, it triggers the release of neurotransmitters into the synaptic cleft. Okay, so these neurotransmitter molecules that have entered the synaptic cleft are able to quickly diffuse through the extracellular fluid that's present in the cleft. And what they do is that they bind to these specialized proteins called receptors that are found in the membrane of the postsynaptic cell, so on the other side of the synapse. So those receptors are depicted here in blue, and the little neurotransmitter molecules are binding to the surface of those receptors. There are different types of receptors that neurons can express. And so one general type is called a ligand-gated ion channel, which is illustrated here. And so here we're again zooming in to a cross-section of the cell membrane. We have the inside of the cell at the bottom, the outside of the cell at the top, and these specialized receptor proteins embedded in the cell membrane. The neurotransmitter molecules that have been released by the presynaptic cell into the synaptic cleft are shown in red here, and they're binding or kind of attaching themselves to the surface of the receptors. And the effect that this has is to cause a change in the structure of the receptor that opens up an ion channel. And then that allows ions that are present in the synaptic cleft, in this case, to then enter the postsynaptic cell. Okay, so these are called ligand-gated ion channels. A ligand in this context is just a molecule that's able to bind and interact with another molecule. And you can compare them to the voltage-gated ion channels that we discussed last week. And so we saw how voltage-gated sodium ion channels, for example, are closed when the neuron is at its resting potential, but then when the voltage of the cell reaches a certain threshold, then the voltage-gated channels open and allow ions to enter the cell. These ligand-gated ion channels are closed when the neurotransmitter molecules are not present, but then when the neurotransmitters are released by the presynaptic neuron and can bind to the outer surface of the channel, it causes them to open. This is just another kind of depiction of how these ligand-gated ion channels change their structure in the presence of neurotransmitter molecules. And so here, this is based off data from a study that was trying to visualize the structure of these channels. And here we're effectively kind of looking from a bird's eye view from the synaptic cleft onto the membrane, and we're seeing like the kind of mouth of the channel, if you like. The red bit is the part of the receptor that's embedded in the cell membrane, and the blue bit is a kind of inner component. And so the gap in the middle is where the ions would like to move through to enter the cell. At the top is when the neurotransmitter molecule is not present. At the bottom is when it is present and it's bound somewhere to the surface of the receptor. And you can see that this produces this obvious structural change in the receptor that opens up the ion channel and then allows ions to enter the cell. Okay, so another general category of receptors that are found in our brain are called metabotropic receptors, which are illustrated here. So what we have here is another type of protein embedded in the cell membrane. We have our neurotransmitter molecules in red, again binding to the surface of that receptor. But this time it doesn't directly open an ion channel in the receptor itself, but rather it causes a structural change in the receptor that then allows it to interact with other chemicals that are found inside the cell. And so this triggers a kind of chain reaction of different chemical reactions that leads to the production of something called second messenger molecules, so just other molecules inside of the cell that are able to move within the cell. And these can move and interact with other things inside the cell, including ion channels. And it can lead to ion channels located elsewhere in the cell membrane opening. So the end result is similar in many ways here, that we have a neurotransmitter binding to the receptor on the outside of the cell, And eventually here, this is leading to ion channels opening in the cell, allowing ions to enter the cell. But it's happening through this less immediate route, this internal pathway of chemical reactions. And for that reason, the effects of these metabotropic receptors are often less immediate, but they can be sort of more long-lasting as well. And so they're referred to as metabotropic receptors because they act via metabolic pathways inside the cell, which refers to this chain of chemical interactions. Okay, and so what does this achieve, this opening of ion channels? It allows ions to enter the postsynaptic neuron, and as we saw last week, the effect of this is then to alter the voltage of the cell, so in this case the postsynaptic cell. So if we have positively charged sodium ions, for example, entering through these ligand-gated ion channels, it will tend to make the cell more positively charged inside. And that depolarization of the postsynaptic cell caused by sodium ions entering through ligand-gated ion channels can then push the voltage of the cell across the threshold that's required to open the voltage-gated sodium ion channels. And like we saw last week, when that happens, it triggers this large influx of sodium into the cell that causes a big spike in the voltage that we refer to as the action potential. Okay, so the key points here are that neurotransmitters bind to receptors that are present in the membrane of the postsynaptic cell. This can cause ligand-gated ion channels to open, which can then depolarize the postsynaptic cell. And if that depolarization reaches the threshold that's required to open voltage-gated ion channels, it can trigger a new action potential to occur in the postsynaptic cell. So in this way, the signal is being passed on, in effect, from one neuron to another. Okay, so are there any questions at this stage? Okay, so a final step in this process is then the removal of neurotransmitters from the synaptic cleft. And so these neurotransmitters have been released by the presynaptic cell. They've kind of filled up the synaptic cleft. They're binding to these receptors and stimulating or depolarizing the postsynaptic cell. And that stimulation ends when the neurotransmitters are broken down or removed from the synaptic cleft. And that can happen in three main ways. The neurotransmitter molecules can just diffuse away. They can float away through the extracellular fluid to somewhere else. They can be broken down, so there are enzymes present in the synaptic cleft that can break down and destroy these neurotransmitter molecules over time. And then they can also be reabsorbed into the axon terminal of the presynaptic cell. And this is a process called reuptake, where neurotransmitters that have been released into the synaptic cleft can be taken up again through specialized channels in the axon terminal so that they can then be reused again in the future. And as those neurotransmitters are released or removed from the synaptic cleft, it means they're no longer binding to the receptors and the ligand-gated ion channels will close. A reason why this final step is interesting is that it's been implicated in the effects of a number of drugs, including psychiatric medications on our mind. And one key example is SSRIs, so selective serotonin reuptake inhibitors, which you'll know are a very commonly prescribed medication for symptoms of depression as well as anxiety. These drugs interact with the function of a neurotransmitter called serotonin, which we'll talk about a bit more later, but is often thought to be involved in regulating our mood, as well, amongst other things. And a part of what they are doing in the brain is blocking the process of reuptake of serotonin back into the axon terminal. So they interact with these channels that would usually reabsorb serotonin back into the axon terminal and block that from happening. And the result of that is that it means that when serotonin is released into the synapse, it will tend to stay there for longer and continue stimulating the postsynaptic cell. Okay, so this medication is effectively facilitating the communication between neurons that's mediated by serotonin. So there's still a lot of debate and research into how these drugs work, and we'll talk a little bit more about that in a later lecture. But in general, these different steps that I've talked about that are involved in communication across the synapse provide a number of different targets that drugs can interact with to affect the ways that our brain functions. Some examples are given here as well for another neurotransmitter called dopamine. So one example is a medication called L-DOPA or levodopa, which is often prescribed for patients with Parkinson's disease as a treatment for motor symptoms of the disease, like tremor and things like that. And levodopa is a chemical that our brain is able to convert into dopamine. And so by ingesting this chemical, we're effectively increasing the supply of dopamine in the brain. Another example is cocaine, which can interact with our brain by, again, blocking the reuptake of dopamine into the axon terminal. And so as I described with serotonin, the effect of that is that when dopamine is released naturally by our neurons, it will tend to stay in the synaptic cleft for longer and have a greater effect on the postsynaptic neurons. So it's facilitating the effects of dopamine. And then one more example are certain antipsychotic drugs that have been developed to treat symptoms of schizophrenia, for example. And these seem to work in part by blocking receptors in the postsynaptic neuron that dopamine would usually bind to. So when these chemicals they themselves can bind with these receptors but not activate them, and then that blocks dopamine from binding to the receptor. And in that way it is inhibiting the sort of usual communication between neurons that relies on dopamine. And so these different drugs can interact with this process in different ways, sometimes facilitating the communication between neurons and sometimes suppressing or down-weighting that communication. Okay, so if you haven't studied much biochemistry before, then one question that might have come into mind is where this specificity that I've talked about comes from. And so I've talked about the idea of there being calcium ion channels in the synapse that can allow calcium ions to enter the axon terminal, but not sodium ion channels. I've talked about the idea of there being serotonin receptors in our brain that serotonin can bind to, but not dopamine. And generally the specificity of these interactions depends on the size, the shape, and the chemical structure of the molecules that are involved. And so you can think of this as being like a kind of lock and key type arrangement where a serotonin receptor will have a binding site that's just the right size, shape, and chemical structure for the serotonin molecule to sort of lock into place and bind with it. but the wrong shape for a dopamine molecule to bind to it. And a reason why that is important is that it's what underlies our ability to develop psychiatric drugs that target particular functions in the brain. So it makes it possible to develop drugs that can interact with the communication between neurons that relies on serotonin while not necessarily influencing other neurons in the brain that are affected by dopamine, for example. Okay, so the key points here are that the effect of neurotransmitters ends when they're removed from the synaptic cleft or broken down, and drugs, medical, recreational drugs, can interact with this process of communication across the synapse in various ways. And and it's really quite amazing that these tiny chemical interactions can actually impact on things like our mood and our alertness and so on. Okay, so we can have a break in a moment, but before we do that, let's do a couple of poll everywhere questions to see how you're following along. Okay, so this first question is to imagine that you take a drug that enters your brain and blocks the calcium ion channels in the axon terminal. The question is, what effect is that likely to have at the synapse? Will it mean that action potentials may fail to propagate into the axon terminal? Action potentials that do propagate into the axon terminal may fail to stimulate the release of neurotransmitter. The postsynaptic neuron may fail to depolarize when neurotransmitters are binding to its postsynaptic receptors, or the reuptake of neurotransmitters may be impaired, leading to neurotransmitters staying in the synaptic cleft for longer. Okay, so it looks like most of you have answered already, and most people have put B, which I agree is meant to be the right answer here. And so we've seen that calcium ion channels have this quite specific role at the synapse, that the calcium ions themselves, when they enter the axon terminal, are able to interact with the synaptic vesicles and cause them to fuse with the membrane of the cell, and in that way release their neurotransmitter. And so what that means is that when the action potential arrives at the axon terminal, if it's not opening these calcium ion channels, then there won't be any release of the neurotransmitter into the synapse. Okay, whereas so the action potential itself, the way that it propagates, relies on these different ion channels, the sodium and potassium ion channels, as we discussed last week. And this reuptake of neurotransmitters also involves channels in the axon terminal, but these are different types of structures compared to the voltage-gated calcium ion channels. Okay, and so one more question is to imagine that a drug is developed that can block the function of all ligand-gated ion channels in the brain. Which of the following do you think might occur if this drug is applied to a neuron? Would neurotransmitters that diffuse across the synaptic cleft be less likely to trigger an action potential in the neuron? Would stimulating the neuron with electrical current be less likely to trigger an action potential in the neuron? Would an action potential that's triggered in the neuron fail to propagate down the length of the neuron's axon? Or would an action potential that arrives at the axon terminal fail to stimulate the release of neurotransmitter? Or do you think it's all of the above? So again, I'll just give you a moment to put in an answer, and feel free to discuss with the person next to you. Okay, so it looks like 2/3 of people put A and about 1/3 put E. And so the correct answer here is meant to be A. And the I guess the trick to the question is that it's specifying ligand-gated ion channels rather than all ion channels. And so we've seen that these ligand-gated ion channels that are found at the synapse and open when neurotransmitters bind to them have this particular role in allowing the neurotransmitters that are released from the presynaptic neuron to depolarize the postsynaptic neuron. Which is why A is correct. The reason why all of the above isn't correct is that these other processes tend to rely more on the voltage-gated ion channels, like what we discussed last week. And so if we stimulated a neuron with electrical current and depolarized the neuron, that could still trigger an action potential in the neuron by causing the opening of voltage-gated sodium ion channels, even if the ligand-gated ion channels are blocked and shut. And similarly, the propagation of an action potential down the length of the neuron, again, depends on the opening and closing of voltage-gated sodium and potassium ion channels. And the release of neurotransmitter is mediated by the opening of these voltage-gated calcium ion channels. So if the question was specifying all ion channels in general, then E would be correct. So any questions? Okay, so let's have a 10-minute break or so and come back at 10:02. And yeah, if you have any other questions, feel free to come up and ask me now. Okay, so we better get started again. All right, so we've been talking about this process where an action potential in one neuron, which is the presynaptic neuron, travels down the length of its axon, triggers the release of neurotransmitters from the axon terminal. Those neurotransmitters can bind to receptors in another neuron, the postsynaptic neuron, and that can lead to ion channels opening in the postsynaptic neuron. Ions, like sodium ions, for example, entering the postsynaptic neuron and then altering the voltage of the postsynaptic neuron. In this case, depolarizing it or making it more positive. And then if that depolarization caused by the opening of ligand-gated ion channels by the neurotransmitters reaches a certain threshold, which in this example is negative 55 millivolts, then it will trigger the opening of voltage-gated sodium ion channels that allow a lot more sodium to enter the cell, causing this much larger depolarization, which is the beginning of the action potential. And in that way, the signal has been passed on from neuron A to neuron B, and that action potential that's been triggered in neuron B may then travel along its axon and then cause release of neurotransmitter onto a third neuron, for example. Another thing that can happen when an action potential occurs in the first neuron is that it triggers the release of neurotransmitter across that synapse again that leads to ligand-gated ion channels opening in the postsynaptic neuron. The influx of ions that produces can cause some depolarization in the postsynaptic neuron, a change in its voltage, but which isn't sufficient to reach the threshold that would open the voltage-gated sodium ion channels. And if that happens, then there will be no action potential in the postsynaptic neuron, and so effectively the signal stops, if you like. It doesn't get passed on through the network of neurons. And so the effect of neurotransmitters on the voltage of the postsynaptic cell when they open the ligand-gated ion channels are sometimes referred to as a graded potential, so meaning a change in the voltage of the postsynaptic cell that can vary in its magnitude. It can be a weaker depolarization or a stronger depolarization, for example. And a key factor that can influence whether the depolarization is strong enough to trigger an action potential in the postsynaptic cell so that the signal gets sort of passed on is the summation of these signals that a neuron is receiving across its synapses. And there are two types of summation. There's temporal summation, where at one specific synapse, so here this is the axon of a presynaptic neuron, this is the cell body of the postsynaptic neuron. So at one particular synapse, there can be multiple action potentials arriving at the axon terminal in rapid succession that can lead to multiple bursts of neurotransmitter, if you like, that can then add up together to cause a stronger depolarization of the postsynaptic neuron. And we'll talk more about that in just a moment. And then that contrasts with spatial summation, which is where a neuron that is receiving signals across multiple synapses, so these are representing the axons of different neurons that are all sending signals to this one postsynaptic neuron. If it receives signals simultaneously, each of those signals might not be enough to trigger an action potential, but together it may cause a strong enough depolarization in the postsynaptic cell to trigger an action potential. And so the idea here is that in the case of spatial summation, at each of those synapses, the release of neurotransmitter and opening of ligand-gated ion channels will cause an influx of ions to the postsynaptic neuron that result in this graded potential, this change in its voltage. Those graded potentials can effectively travel across the cell body of the neuron and add up together at the axon hillock. So that point of connection between the axon and the cell body of the neuron, where there's a high concentration of these voltage-gated sodium ion channels. And so if together these graded potentials can depolarize the axon hillock to a sufficient extent, that can then trigger an action potential in the axon hillock that then travels down the length of the neuron's axon. Okay, so that's the basic idea behind the summation of synaptic signals. Another way of illustrating that might be helpful is with a plot like this, where on the left here, first of all, we're representing one graded potential, which here is labeled as an excitatory postsynaptic potential. So it's excitatory because it's depolarizing the postsynaptic neuron. It's moving the voltage of the postsynaptic neuron closer to the threshold that's required to open the voltage-gated sodium ion channels and trigger an action potential in that neuron. In the case of temporal summation, if we imagine that we have 2 or 3 action potentials occurring in rapid succession in the presynaptic neuron, so we're just focusing on one synapse here, if each of those action potentials generates a small burst of neurotransmitter that opens some number of ligand-gated ion channels in the postsynaptic cell, it might be that each burst of neurotransmitter is only capable of producing a small depolarization in the postsynaptic cell, but when you have 2 or 3 arriving in quick succession, the change in voltage adds up and may push the voltage across the threshold that's required to open voltage-gated sodium ion channels and trigger an action potential in that postsynaptic cell. So that's the temporal summation of excitatory postsynaptic potentials. In the case of spatial summation, it's a similar idea that in general, neurons are not just connecting one-to-one. Generally, each neuron will be receiving signals from many other neurons. In this case, it's receiving signals from 3 different neurons. And again, the signal that the effect on the postsynaptic cell that occurs when neurotransmitter is released from one of those synapses might only be enough to cause a small depolarization in the postsynaptic neuron. But if all 3 synapses are releasing neurotransmitter together, the effect can add up to produce a stronger depolarization that may reach the threshold required to open voltage-gated sodium ion channels and then trigger an action potential in the postsynaptic neuron. One other complication is that these signals that neurons receive can also be inhibitory as well as excitatory. And so an inhibitory postsynaptic potential is shown on the right here. And it's referred to as being inhibitory because it's moving the voltage of the cell further away from the threshold that's required to trigger an action potential. So it's hyperpolarizing the cell rather than depolarizing it. In this case, it's making the cell more negatively charged inside rather than more positively charged. And as a result, that decreases the likelihood of the neuron being able to generate an action potential. So generally, we can define a synapse as being either excitatory or inhibitory. So at an excitatory synapse, an action potential in the presynaptic cell is depolarizing the membrane of the postsynaptic cell, making it more likely that the postsynaptic cell will produce its own action potential. And commonly that depolarization is caused by ligand-gated ion channels opening that allow these positively charged sodium ions to enter the postsynaptic cell. So making it more positively charged inside. At an inhibitory synapse, an action potential in the presynaptic cell is hyperpolarizing the postsynaptic cell, so making it less likely to produce its own action potential. And this is typically caused by ligand-gated ion channels being opened by the neurotransmitter that allow chloride ions to enter the postsynaptic cell. So chloride ions we haven't touched on previously, but they're another type of ions that are present in the extracellular fluid, and they're negatively charged. So as they enter the postsynaptic cell, they're making the cell more negative inside. So the dendrites of any particular neuron can synapse with axons of many other neurons, and some of those will likely be excitatory and others inhibitory synapses. And so whether a neuron is active, whether it's firing action potentials or not, depends on the mixture of excitatory and inhibitory signals that it's currently receiving. And this leads us to the idea of neurons being able to wire together or connect together into networks or circuits. So to illustrate this idea, we're going to use a sort of abstract way of depicting how neurons interact with each other. And so here, these yellow bubbles are representing 3 different neurons, neuron A, B, and X. And the red arrows here are representing the effects that neurons have on each other across the synapse that they share. So in this example, we're going to say that neuron X has a threshold of 1, so it needs an excitatory input of 1 in order to become active, to produce an action potential. neuron A and B, when they produce action potentials, are going to give neuron X an excitatory input of 1 each. So the question here is what does it mean if neuron X is active, producing an action potential? And we can illustrate it like this. So if both A and B are silent, they're not giving any input to neuron X, then neuron X will also be non-active. If both A and B are active, then they'll both be giving an input of magnitude 1. So overall, neuron X will be getting an input of 2, and that exceeds its threshold of 1, and so neuron X will be active. If only A is active or only B is active, then again, neuron X will still be getting enough input to trigger an action potential. And so in this example, activity in neuron X is signaling something about the inputs that it's receiving. So it's signaling that at least one of A or B are active. If we then make a simple change to the network, so we just increase the threshold of neuron X to 2, then it comes to represent something different about the inputs that it's receiving, because now if only A is active or only B is active, then the input of 1 that each of those neurons provides is not sufficient to trigger activity in neuron X. And so in this case, neuron X will only be active if both A and B are stimulating it at the same time. And so now activity in neuron X signals that both A and B are active. And so in this sense, the activity of neuron A is representing a kind of information about the inputs that it's currently receiving. And these are very simple and very abstract examples, but as we go through the course, we'll see how this kind of wiring between neurons is useful for understanding how the brain is implementing specific functions of our mind. So in the case of vision, for example, this type of wiring between neurons can mean that the activity of neurons comes to represent meaningful information about the signals that our brain is receiving from our eyes, and that this allows us to do things like recognize faces and recognize objects around us. If we make the network slightly more complex, we just add an extra component, neuron C, and C is now going to send an inhibitory input of 1 to neuron X. Now the conditions under which neuron X suddenly become quite a bit more complicated. And so the details are not really important here. The point is that as we kind of expand the network of neurons that we're talking about, and in the brain, neurons can receive signals from hundreds or thousands of other neurons at once. The behavior of a neuron in terms of when it's active and what that represents about the inputs that it's receiving can become quite complex. Okay, so to illustrate a little bit further some of the concepts here, we can do a couple more poll everywhere questions. And so in this first question, we're going to consider this same wiring diagram again of neurons A, B, and C sending signals to neuron X. And again, the number next to each of the red arrows is representing the strength of the signal and the type of signal that neuron X is receiving across each of these 3 synapses. And so the question is, which of the following factors do you think could affect this number? Is it the number of ligand-gated receptors that are found in the postsynaptic membrane of neuron X? The number of neurotransmitters that's released by these presynaptic neurons, A, B, and C. The speed of neurotransmitter reabsorption into the synaptic cleft. Whether the neurotransmitters released are binding to sodium or chloride ion channels, or do you think that all of these factors could influence this number? Okay, so most of you said E. A fair few said D as well, and so the correct answer here is meant to be to E. That the magnitude of this number can be influenced by this sort of different variety of underlying biological processes. So, for example, if you have if there are more ligand-gated receptors found at the postsynaptic neuron of a particular synapse, it means there's more opportunities for neurotransmitters to bind, open more channels, and cause more ions to enter the postsynaptic cell and depolarize it to a greater extent. If there's more neurotransmitters being released across a particular synapse, again, that can provide more opportunity for binding to more of the ion channels and opening more of them and causing greater depolarization of the postsynaptic cell. I gave the example earlier of how the reabsorption of neurotransmitters at the synaptic cleft can, and the speed at which that occurs, can affect how much the presynaptic neuron stimulates the postsynaptic neuron. And D, whether the neurotransmitters opening sodium or chloride ion channels affects the polarity, whether it's a positive or negative number, whether the entry of those ions are depolarizing or hyperpolarizing the postsynaptic cell. So for those of you who put D as an answer, that also makes sense to me, because in this particular example, what's differing between the neurons is just the sign, and that factor is what's affected by D. Okay. Okay, so one other example we can sort of think about with another poll everywhere question. We're going to consider this simple network where we have 3 neurons, so neuron 1, 2, and 3. Neuron 1 is forming excitatory synapses with both neuron 2 and neuron 3. Neuron 2 is forming an inhibitory synapse with neuron 3. The question here is, what do you think would happen to neuron 3 when neuron 1 produces an action potential? Do you think there'll be no change in the voltage of neuron 3? Do you think there'll be brief excitation of neuron 3? Hyperpolarization of neuron 3? Or do you think it's impossible to say based off these details? Okay, so the responses are split a bit between B and D, and what this question is meant to help illustrate is one of the complexities that occurs when neurons wire together, and in particular that it can affect the timing of effects on the postsynaptic neuron. And so in this example, if neuron 1 is producing an action potential, this will cause excitation of both neuron 2 and neuron 3, and then excitation of neuron 2 may trigger an action potential that then causes inhibition of neuron 3, but that inhibition will likely be occurring a little bit later than the initial excitation of neuron 3, and that's because the signal, if you like, needs to pass across the synapse with neuron 2 and then another synapse with neuron 3. Okay, so you may get an initial excitation of neuron 3 through this synapse, and then slightly later than that, an inhibition of the neuron. And so it makes sense that B can be an answer here. So you get this brief excitation of the dendrite that's then sort of cut off by the brief because it's cut off by the subsequent inhibition. D is also a fair enough answer as well, that I haven't told you anything about like the strength of the synapses here, for example, and so that might be important for really predicting what's happening. So yeah, so really either of those answers is reasonable here. Okay, so any questions at this stage? Okay, so the key points in this section is that these inputs that a neuron receives can be both spatially and temporally summed together to activate or to determine what happens in the postsynaptic neuron, whether it produces an action potential or not. Excitation and inhibition are these different effects that neurons can have on other neurons, and they can interact in complex ways, like, for example, influencing the sort of timing of events. And neurons are wired together into circuits or networks that can process signals in complex ways. And so here we've just seen some kind of simple and abstract examples, but as we go through the course, we'll see how the wiring between neurons can contribute to perception and other cognitive processes. Okay, so these synaptic inputs that a neuron can receive, excitation and inhibition, can vary in their magnitude. So you can have a sort of a weaker excitatory postsynaptic potential or a stronger one, as we've been discussing. But the action potential itself, this much greater depolarization that occurs when voltage-gated sodium ion channels open, is a all or nothing event. So it either happens or it doesn't. The actual magnitude of the action potential itself is not something that varies typically within a given neuron. And that's because of its dependence on the voltage threshold of these voltage-gated sodium ion channels. So either the depolarization of the neuron is lower, doesn't reach the threshold and can't trigger the action potential, or it does reach the threshold and then you get a full action potential as the voltage-gated channels open. But what can vary in a neuron's activity, depending on how much excitatory input it's receiving, is the rate at which it's producing these action potentials. So often when we talk about a brain area becoming more active, for example, what we mean is that the neurons there are producing these action potentials at a faster rate, a greater frequency than they were before. So last week I gave you this example and showed you these videos from this really influential study by Hubel and Wiesel, where they were recording the electrical activities of neurons in the cortex of cats while the cats were being shown different images on a screen. Some of the data from their study is illustrated here. So what this is representing is that when the cat is being shown a horizontal line, they're not detecting any action potentials in the neuron that they're recording from. When they rotate the line to a diagonal line, the neuron that they're recording from starts producing some action potentials, and then when they show the cat a vertical line, in this example, this particular neuron starts producing a faster rate of action potentials. And so you can think of the rate of action potentials as being a bit like the language of the brain, or the code that neurons are using to communicate with each other. That in this example, the rate of action potentials is representing something about what the cat is seeing. A faster rate occurs when the cat is seeing a vertical line, a slower rate when it's seeing a diagonal line. So the key point here is that action potentials are all or nothing in terms of their magnitude, but greater excitatory input to a neuron can increase the rate of these action potentials. Okay, so to finish up with, we can talk a little bit more about neurotransmitters, so these chemicals that are released by neurons and can have an effect on the function of other neurons. I've already alluded to how there are different types of neurotransmitters in our brain, like serotonin and dopamine, for example, and different neurons in our brain will tend to release different neurotransmitters. Two neurotransmitters that you should be aware of that are called glutamate and GABA. And so GABA stands for gamma-aminobutyric acid. And these are the primary excitatory and inhibitory neurotransmitters that are found in our brain. And so glutamate usually works as an excitatory neurotransmitter. It can bind to ligand-gated sodium ion channels in the postsynaptic neuron, causing positively charged sodium ions to enter the postsynaptic neuron and depolarizing the neuron. Whereas GABA tends to act as an inhibitory neurotransmitter. It also binds to ligand-gated ion channels, but these are ion channels that allow chloride ions to enter the postsynaptic neuron. And these negatively charged chloride ions, as they enter the neuron, they hyperpolarize the cell. They make it less likely to produce an action potential. There's also a class of neurotransmitters that are referred to as neuromodulators, and that includes serotonin and dopamine that we've mentioned already. A key difference between neuromodulators and other kind of classic neurotransmitters is that neuromodulators often act via the metabotropic receptors rather than the ligand-gated ion channels. And so glutamate and GABA generally binding to these ligand-gated ion channels and causing an immediate response in the postsynaptic cell by opening the channels and allowing ions to flow into the cell. These neuromodulators are usually binding to these metabotropic receptors that aren't ion channels themselves, but rather can trigger chemical reactions inside the cell that can have a more gradual, but in some cases a longer lasting effect on the cell's behavior. And these neuromodulators also tend to influence brain function in quite a diffuse manner. So to give you just a few illustrative examples, one neuromodulator is called noradrenaline, and it's a chemical which is produced in our brain in a particular structure called the locus coeruleus, which is this small cluster of neurons that's found deep in our brain. But those neurons send out their axons all over our brain, so all over the cortex and also the cerebellum, for example, and release noradrenaline kind of widely across the brain. And noradrenaline is thought to be involved in regulating our kind of mental arousal or mental alertness, so like waking up our brain, for example. And so it makes sense to think of it as a neuromodulator, that it's something that's sort of having a wide, diffuse effect on the brain, which parallels with our experience of mental alertness as kind of like a general upregulation in the function of our brain. So the details of the anatomy here you shouldn't worry about too much. So these are just kind of illustrative examples. And next week we'll have a lecture on anatomy where I'll make it a bit clearer what's worth knowing in terms of the anatomy of the brain. Another example is serotonin, this neuromodulator that's, again, produced in these small structures deep in our brain called the raphe nuclei. And again, the neurons in this area that produce serotonin send their axons widely across the brain and release serotonin to different areas of the brain. And as we saw in the example of how antidepressant drugs can interact with this system, serotonin is thought to be involved in the regulation of mood and other functions like sleep as well. And then finally, dopamine. There's a common theme here. It's produced in these structures, small structures in our brain, found deep in the brain. In this case, they're called the substantia nigra and the ventral tegmental area. And these send out their axons to different parts of our cortex and other structures in our brain below the cortex. And its function is still being debated in various ways, but it's often thought to be involved in our experience of reward or pleasure, the regulation of motor function, our movement, and maybe other aspects of our cognition. And so, for example, in Parkinson's disease, there seems to be this specific loss over time, kind of death of neurons in the substantia nigra region of the brain. These neurons sort of producing dopamine, and the sort of often the earliest sign of that are these impairments in motor function, so tremors and difficulties with walking and things like that. Okay, so what are neurotransmitters? They're chemicals, and often they're amino acids or related to amino acids. So amino acids are these category of chemicals that are found very widely in our body, and they're the building blocks of proteins. So each protein in our body is made up of a string of different amino acids. And so glutamate and GABA, these neurotransmitters, are themselves a type of amino acid, and serotonin, dopamine, and so on are forms of amino acids that have been modified in our body. And so when we eat food that contains protein, our body is breaking down the protein, extracting these amino acids, and using that for various things in our body, including as molecules that help neurons communicate with each other. One example shown here is an amino acid called tryptophan, which apparently is found in kind of various foods that we eat, like eggs and milk and fish and certain types of vegetables. And our body is able to adjust the chemical structure of tryptophan to create serotonin. And then this serotonin is used in our brain to help neurons communicate with each other. And so this process where these neurotransmitters are being synthesized, in this case from tryptophan, is often happening within the neurons that will actually use that neurotransmitter. Okay, so the key points here are that glutamate and GABA are acting respectively as the primary excitatory and inhibitory neurotransmitters in our brain. And then we also have these neuromodulatory systems that include neurotransmitters like noradrenaline and serotonin that are able to modulate the activity of our brain in a more diffuse manner. So any questions? Okay, so if you have any questions, you're welcome to come up and ask me now. Next week we'll be moving beyond the level of individual cells in our brain and begin by looking at the general anatomy of our brain. All right, thanks. I'll see you next week.